For our first speaker, I would like to introduce Howard A. Bern. Howard Bern is a tumor and comparative endocrinologist at the University of California in Berkeley in the Department of Integrative Biology and Cancer Research Laboratory, where he is Professor of Integrative Biology. Dr. Bern has been working in the department for the past 47 years and has served as the chair of the group in endocrinology for much of that time. He is a member of the National Academy of Sciences, the American Academy of Letters and Science, and academies in India and Italy. Dr. Bern has served as visiting professor at more than 10 universities around the world. Dr. Bern received his doctorate from UCLA and has honorary doctoral degrees from the University of Rouen in France and from Hokkaido University in Japan. He is the author and co-author of more than 500 scientific papers and is the author and co-editor of 6 books. I'd like to point out to you in your program today that the article that he had written, The Fragile Fetus, and I would highly recommend that you take the opportunity to read that. Dr. Bern's research has focused on hormones and cancer, on the long-term effects of early exposure to hormones, on neurohormones, and on the hormones in fishes. It is his interest in the long-term effects of early exposure to hormones that led to his concern with the human diethylstilbestrol, DES problem, which he is going to discuss with you today. So please welcome Dr. Howard A. Bern.
I think before starting, I'd like to make I have 3 preliminary remarks. The first is to express my admiration for Brenda Edelman and her committee and her co-organizers for this community-based program. In my opinion, this is where effective action is generated. I think it's apparent that that's so. So my compliments to you. Then I want to say something about Lou Gillette. Lou Gillette leaves me in a rather odd position. There is someone who looks older than I do, who refers to me as his grandfather. Excuse me, Buzz. All I can say is that his comments have an aging effect on me. Another comment is that there is something that goes on in these kinds of affairs in which the speakers this morning and I and John McLaughlin have engaged in over some years. It's kind of what I would call progressive robbery of material. So that we leave John McLaughlin little to talk. And what truly amazes me is that he still talks for an hour anyway. Enough levity. The subject is serious. My purpose is to discuss the background of experience that adds to the alarm many of us share about the presence of estrogens in the environment. One needs to recognize that early exposure to hormones and to other agents may have long-term and permanent effects upon the organism. The issue here is early exposure, long-term and permanent irreversible effects. If pregnant mammals are treated with various drugs and hormones, it has been customary to say, well, they must be okay because there are no obvious birth defects. This is not the tolbutamide story. This is a story where there may not be birth effects, but there may be long-term permanent effects nevertheless. The issue of birth defects is simply not the issue. Now, some years ago, 1960 in fact, we began studies on laboratory mice when my first Japanese postdoctoral fellow, Noboru Takasugi, came to my lab in Berkeley. As I say, he was my first postdoctoral. He just retired as president of Yokohama City University. And at that time, as a result of his research, as a result of the research of Thelma Dunn and her colleague at the National Cancer Institute, and as a result of research by Albert Reynaud in Paris, we were cautioned on the danger of early exposure to excess amounts of sex hormones during fetal life. So all 3 different laboratories called this to the attention of the scientific public and hopefully the reading scientific public. But you wouldn't know this from the response. What happened in mice, as an example, was that early exposure to female hormone, and what one can use in the case of the mouse is a newborn mouse, a neonatal mouse, because reproductive development has not been completed at the time of birth. And in the female mouse, one witnessed a very high incidence of persistent vaginal overgrowth, which can lead to vaginal cancers. And this was apparent in at least in 2 of those early studies. 10 years later, in humans, see, I can run this. Yeah. Here is the laboratory mouse, which is a very amiable animal. And in humans, as I said, 10 years later, Arthur Herbst was co-author of this very important paper on adenocarcinoma, which we now would call clear cell cancer, in the vagina of young women. If you will read this abstract, and you don't have to, you would find no mention of what the possible cause was. It took another year before it was recognized that the mothers of these women had taken a synthetic estrogen, diethylstilbestrol, very potent estrogen, while they were pregnant, and indeed during the first 3 months of their pregnancy. The reason that these mothers took this hormone had to do with ads such as this, which indicated that DES would prevent all kinds of things which incidentally it did not prevent, and recommended it for routine prophylaxis in all pregnancies. The basis was, well, even if you're not having any trouble, this is a harmless drug and it might do you some good. There were no birth defects observable after its usage. Well, let me tell you a little bit about this phenomenon as it developed. The exposure period when women were taking this drug was approximately 30 years, about 1940 to 1971, when it was declared off limits for this kind of usage. There has been a debate about whether the statistics that indicate the association between DES exposure during pregnancy and the occurrence of this clear cell cancer in offspring, whether they were really meaningful or not. It has been figured that maybe 5 million or more DES offspring are in the United States. The figure may be higher. It's very hard to conclude. The figure could even be lower by, say, 1 million or so. What was interesting about the American epidemiological statistics was that there was one other small country which used DES in the same manner and for the same length of time as we did, and this is the Netherlands. And the statistics from the Netherlands and the statistics from the United States are exactly the same. And in those countries where DES was not used, the incidence of clear cell cancer of the vagina in young women is 0 or close to it. The risk of vaginal cancer for these DES daughters has been calculated as being between 1 in 1,000 to 1 in 10,000. That means it's a relatively rare cancer, but I think anyone will agree that one case is too many. This form of vaginal cancer had been, up till this time, seen only in older women, in postmenopausal women. It had not been reported in any young women. Now, in addition to the United States and the Netherlands, the drug was used extensively in Canada, in Australia, in France, in Spain, and undoubtedly other countries. In the United States, Kenneth Knoller calculated a few years ago that 7 to 10% of women in the United States in the age group of 24 to 40 years had been exposed in utero to DES. This is a very large population, and what we are talking about is really a massive, unintended experiment on the human population, which led to the recognition of what I said to begin with, long-term permanent effects of early exposure to a hormone. Now, the development of cancer in a small number, relatively, of DES daughters is not the only consequence of early exposure to DES. This is the target of DES in our own species. This is a human female pregnant. The real target is the fetus that she carries, and this is about a 3-month-old, end of the first trimester fetus, and exposure before that time would lead, in some cases, to vaginal cancer. A group called DES Action and its leaders, Pat and Nora Cody, and up until relatively recently, Judy Turiel, are here among you, so you can get much information from them. But this group, which has done marvelous things in the area of making not just the ordinary citizens of the United States aware of the DES problem, they've made legislators and directors of national cancer institutes aware, and that's much more difficult. A year or so ago, they produced a table which summarized the adverse reproductive effects of DES exposure in utero. So this is the kind of things that one could encounter in DES daughters and even effects upon DES sons. And maybe this list is being extended at the present time as more recognition of pathology in the human male is emerging. I think you should maybe, in addition now to the occurrence of cancer, should look at this area, for example, of adverse pregnancy outcome. The next slide illustrates what this means. In fact, this is the kind of uterus that DES daughters show, where the lumen is T-shaped and the structure of the uterine body is such as to indicate truly abnormal form. The muscle is really poorly developed, and this leads to a decrease in successful pregnancy outcome. And Arthur Haney, who has studied this in detail, feels that this phenomenon, the T-shaped uterus, is a characteristic of all DES daughters, not just the very small percentage that show vaginal cancer. So this is what DES action, I can't go backwards, yeah. This is what DES action meant by one of the adverse reproductive effects, quite apart from the cancer issue. Lou Gillette has already referred to the fact that the developing embryo, and this is a good picture of the developing mammalian, including human embryo, but in fact the developing vertebrate embryo, passes through an indifferent stage. I don't really like that term. The implication is that the developing organism doesn't really care what's about to happen. I think the investigator was indifferent, but had more of a sense of commitment. In general, develops either into a female, develops a female reproductive tract or a male reproductive tract, though intersexual events are obviously common enough and will occur with greater frequency upon exposure to hormone disruptors of one kind or another, endocrine disruptors. And what may occur in both females or males is retention in one area or another of some parts of this system that characterizes the female or characterizes in the male only in the opposite sex. And this retention could be the source of a variety of disorders in humans. And the pathological explanation for some of the changes that occur in humans involves this notion of abnormal duct retention. Indeed, it has been suggested that the clear cell cancer in the human DES daughter arises ultimately from the fact that some of this duct system, which you see here in the developing embryo, which later retreats from the vagina, instead persists in the vagina and becomes the precancerous bed from which the cancer can arise. Now, we want to emphasize for the next period of time the importance of the newborn mouse, that's what that is, the neonatal mouse as a model for studying and analyzing what we'll just call the DES syndrome, the long-term effects of early exposure to estrogens in this case. And this is a very important model. John McLaughlin's model initially, which involved feeding DES in the diet to pregnant mice in the latter part of pregnancy and continuing this feeding to the young after they were born, is a similarly useful source of information. I want to emphasize that there's one aspect of exposure to the agents that we are about to discuss that you have to keep in mind, and that is that this exposure is not just something that can occur anytime, but rather occurs during what we call a critical period. No case of vaginal cancer in DES daughters has been reported if exposure to DES began after the first 3 months of pregnancy. So the critical period is the first trimester in the case of the human DES daughter. In the case of the mouse, the neonatal mouse, the persistent vaginal changes, which are comparable in part to what is seen in the human species, will only occur if the mouse is treated, or rather maltreated, within the first 3 or 4 days after birth. That again is a critical period. For the development of the clear cell cancer in DES daughters, Herbst early suggested, and it's certainly borne out by reality, that it must be the hormones of adolescence, hormones of puberty, if you will, that contribute to the ultimate emergence of the clear cell carcinoma. That the cancer has never shown up in a pre-pubertal female, but one which has been under the influence of hormones associated with adolescence. In mice, we have also found that if you have a neonatally exposed mouse, then treated later in adult life with estrogen, that the incidence and severity of the lesions that occur are intensified. It's a comparable kind of situation. Now, the mouse model has given us a large variety of information, and I have no intention of discussing this variety of information in detail, but maybe its utility is best indicated by my revision and, of course, improvement of this table from the McLaughlin Laboratory, which listed phenomena occurring in both the female and the male offspring as a result of early exposure to DES, and in both the male and female are a good number of changes which are quite parallel. Other changes besides the ones I've discussed have been seen. Lou Gillette this morning discussed polyovular follicles, so I don't have to show my slide of that, which is certainly better than his. But that is a change which occurs in mice, and it has even been suggested that this kind of polyovular situation may occur in human development when there are abnormal hormonal situations. I think at this point it is important to indicate to you that, and I don't want to go into this in detail, that really DES, when it is given, can act in two separate ways. One way is that it can have a direct effect on the vagina during the development of the vagina. Another way is that it could act through the brain, pituitary, and ovary to result in the continued secretion of estrogen during adult life, so that the estrogen supports the emergence of these vaginal changes. The differences are that if you take the ovary out of a young mouse, the DES effect that it has had is permanent. If you take the ovary out here, then the changes disappear because they are dependent upon this continued supply of estrogen. And I think it's important then to say that agents such as DES and the ones we're later going to discuss can act as hormone mimetics in regard to inducing changes, or it can act by disrupting the endocrine system, which ends up in the production of an agent that influences, is responsible for the changes that are seen. And I like to make this distinction, which I think semantically one could argue against, nevertheless, between estrogen mimetic and endocrine disruptive. Now, there are many reproductive and non-reproductive effects of early exposure to DES and to other agents on mice. And I made this chart some time ago. I thought of making it more complicated by giving you more arrows to forget about. But I think this is enough to indicate clearly that so far as the mouse is concerned, every part of the female reproductive system and the male reproductive system can be influenced by this early exposure. In the case of mice, this is also true of the mammary gland, of the breast. There are no data at the present time that indicates serious changes in the breast or an increased incidence of breast cancer in DES daughters. But the potential certainly existed. Then there are effects on the neuroendocrine system, there are effects on the immune system, and I consider that to be something that deserves comment. I had a PhD student some years ago, co-supervised by my colleague Phyllis Blair and myself. This is Dr. Susan Ways, and Susan Ways wrote her PhD thesis on the immunological effects, actually immunodepression, which occurred in mice that were exposed neonatally to estrogen. She later went on and got her medical degree, and the first paper she wrote as an MD cum PhD was a study on a human population which indicated that DES daughters show significant immunodepression. In this situation, again, the mouse essentially predicted what was ultimately found in the human. Another area which deserves comment, that if there are effects on the nervous system, it's not surprising there would be behavioral ramifications. And this is a difficult area with which to deal. In mice, there is no question, but sex behavior is changed radically by this early exposure to estrogen. So DES daughters have been studied in a variety of ways to determine whether DES exposure caused any psychological changes. Various things have been reported, including changes in judged by cognitive tests, intelligence tests, skill tests. What they actually mean, I'm not sure, but there appear to be some significant differences between DES daughters and a suitably chosen control group. A more recent study indicates that the incidence of lesbianism among DES daughters is significantly higher than it is in a controlled population. I don't really respond much to that kind of a claim because if you consider what it does to the DES daughter to know that she is a DES daughter, or what it will do to the DES son if he finds out that he is a DES son, then it would be a little difficult to distinguish between behavioral effects of the DES itself and the fact that the individual being studied is already threatened, if you will, by knowing that they are suffering, in this case, reproductive changes as a result of the DES exposure. This is a problem in dealing with the psychological consequences of early hormone exposure. I might add to this and say that effects on bone growth development have also been reported, and effects on one tiny little muscle have also been reported. So this is not the end of this picture. I'll try next time I talk about this to have a newer slide. I think it's important again to point out that the developmental effects of DES have been recognize both in experimental animals and in humans, and indeed the leading clinical expert in this area, plus I, that's right, I guess, have been able to collaborate sufficiently to produce a book that has both human and experimental data. But that book is old, still good, but it's a good 14 years old. There is a more recent book where the editors were two Japanese scientists, both of whom spent time in my Berkeley lab, I'm pleased to say, where again the data are brought up to date, at least as of 1988. There's a newer book by Knoller, which has put together the clinical data, but for reasons that I won't explain, namely that I couldn't deliver my chapter on time, ignores most of the experimental data. But Knoller's book does point out something very important, that there are increases in abnormality, there are abnormal changes not at all related to the reproductive system, which one can find significantly in DES daughters. An increase in the occurrence of autoimmune diseases, an increase in the occurrence of eating disorders, anorexia, bulimia, and again, one wonders how much the insecurity of being a DES daughter contributes to these situations, and various other infectious diseases which are found in higher percentages in DES daughters. I think at this time I have to raise another problem having to do with the DES offspring, and that is that the oldest DES daughter is probably about 40 to 50 years old. And the primary dangerous problem for DES daughters is the issue of clear cell vaginal cancer. This is a disease of older women. So one of the fears that students in this area have had has to do with whether there will be a second surge of problems, reproductive and otherwise, that will turn up in DES daughters as they go through the menopause and become older. This is just simply to raise for you the full spectrum of possibilities in regards to this situation. I'd now like to say something about other estrogens. We've talked about natural estrogens because the early experiments done, for example, in my lab, involved the use of ovarian estrogen. We've talked about synthetic estrogens like DES. We should say something now about plant estrogens and their effects, and there are many of them. Plant estrogens are very interesting because they're found in such in grains, mostly due to a mold which grows upon the grain and produces zearalenone, which is one of the plant estrogens. Seedlings of various plants, alfalfa sprouts, which I detest anyway, soy products, including tofu, which I like very much. It is quite possible that this presence of phytoestrogens, plant estrogens, is even beneficial to our own species in what I'll say are possibly moderate amounts. This has been claimed in regards to the lower incidence of prostate cancer in Japanese men as compared with Western men. It has been claimed in other areas as well. Maybe Dr. Moses will discuss this. But I want to assure you that these agents in adults can be very negative in their effects. In Australia, where subterranean clover, I think, was imported as a possible fodder for cattle. The effects of engulfing the estrogen in the subterranean clover was to induce sterility, and there is even a report in Nature of some years ago called nymphomania in cows, something which I have given some time to internal visualization. And no matter how I look at it, I'm horrified. There are plant estrogens, you will be sorry to hear, in hops and therefore in beer, in rice, therefore in sake, and in corn, therefore in bourbon, including Jack Daniels, which I drink regularly. I am not proposing that one give up eating those foods or drinking those liquors. I do, however, want to indicate the potential problems that can arise. And this all has to do with adult effects. When developmental effects are studied, as my former student, Dr. Cynthia Burrows, has studied the effects of cumestrol. Indeed, in my lab, we've also studied the effects of zearalenone. These are very weak estrogens by adult criteria. But when they were given to developing mice, the effects that they had were very similar to what one sees with natural and synthetic estrogens. And the same point, incidentally, can be made about anti-estrogens. If the tamoxifen and clomiphene are given to neonatal mice, they induce the same battery of pathological changes that were seen after, for example, DES administration. So this may be an anti-estrogen largely in the adult mouse, but in the developing mouse, it is not seen as such. It is seen as an estrogenic agent, and I think that is very important. This possibility of effects has even been demonstrated in the human by Professor Cunha at San Francisco, UCSF, who did experiments on cultured human urogenital tracts on babies that were either aborted or were born prematurely, and found that clomiphene and tamoxifen had identical effects upon these developing tracts as did DES. So this, the mouse data that I mentioned initially, can be extended even to the human species. And then we have the problem of environmental agents, which everyone has been discussing, and which I would like to say a little bit about. This is an abbreviated list. It's to be impressed upon you that many of these are organochlorines, and they have stimulated the most attention, but that the alkophenols are important. Other agents, including phthalates, as was mentioned this morning, may be significant on this list of endocrine disruptive agents. One has to keep in mind that these agents may act as estrogens, but may also act as anti-estrogens, may be anti-androgens against the male sex hormone, and may be active as anti-thyroid agents. Anti-thyroid agents. Let me comment upon this because you heard a little this morning from Shepherd Bliss about the possible problem of the disappearing amphibian. The disappearing amphibian is a real phenomenon, and we don't know why they are disappearing. Exposure in the environment during the larval or tadpole stage resulted in 100% females. Now, if the amphibian doesn't metamorphose, it doesn't promise very much for the continuation of the species. And if they are 100% females, it also does not suggest that the species will persist. So the disappearing amphibian problem is not to be taken lightly, and it is possible that environmental estrogens or endocrine disruptive agents are only one possible cause, but nevertheless they ought to be considered. This is illustrative from a recent issue of Science about the PCBs packing a hormonal punch. But the punch that is dealt with here is the fact that they are acting as anti-thyroid agents. And we know also that they can act as estrogen or anti-estrogen as well. This focus on organochlorines is indicated by the fact that even Science magazine recognized that this was real, although if you read the article you might come to the conclusion that it really wasn't very dangerous. Other organizations like Greenpeace were a little bit more definite in their opposition to the use of organic. I had to make sure I used the right term. Now, one of the things that we talked about earlier, was discussed earlier, was the real need to have some tests which would distinguish developmental effects, which would allow you to say about an agent, not that it acted as an estrogen in the adult, for example, by inducing vitellogenin, as was discussed, but rather that it had a developmental effect. And this gets to be very disturbing to people like in the EPA who have to face up to this kind of testing, because it all looks as if these were very long-term tests. And my colleague and former postdoc, Professor Iguchi, and I put this list together. I don't know which one of us stepped on the slide, but somebody put this list together to indicate that there are tests available involving the female neonatal mouse, which take just a matter of a few days, or maybe a month, that you don't have to study these for the lifetime of the animal. Now, work in regards to the developmental effects of such agents as PCBs is underway. It is being studied by Fonsal, by Lovell Jones, and by Professor Iguchi himself. So, if I may use this slide to indicate that the endocrine disruptor can not only affect the adult and the developing organism, juvenile, but can have profound effects upon the embryo and during fetal development, and also can be transmitted, transported to the egg, remembering that some of these agents are held within fat within the body, within lipid deposits, and may stay there for years and years, and can be released and transferred to the egg of non-mammalian vertebrates. The endocrine disruption can occur at any level of development, and the adult containing an endocrine disruptor can influence fetal development, because that's what the DES phenomenon is all about. The adult has taken in DES and has transplacentally passed it on to the fetus. And in non-mammalian vertebrates can pass these agents on to the egg, where they are stored in the egg yolk, and can, if they are acting in an endocrine disruptive fashion, create a fair amount of havoc. Time is getting short. I did have one other topic which I'll not deal with, to try to show you what evidence we have for better or worse, that the environment is estrogenic and maybe increasingly estrogenic, but I think this is derivable from the discussions that you have heard already. I'd like to conclude by just underlining the points that I think need to be remembered from this historical experience of working either with DES offspring or with the mouse model for the DES syndrome. Here are the points. Exposure during development may lead to long-term permanent changes in a variety of body systems, not just the reproductive system, including cancer. That these changes may not at all be visible as birth defects. That effective exposure may occur only if the exposure occurs during a critical period, which may be very short or may be fairly extensive. That estrogens are indeed estrogens by adult tests, but testing for developmental effects is essential, should be a part of whatever protocol agencies use, such as the EPA or the FDA. You have to keep in mind that the fetus may not see estrogens in the same way as the adult sees estrogens. Agents which are not particularly estrogenic in the adult may be seen as potent estrogens by the developing organism. These effects that we have been talking about are epigenetic. Testing usually looks for genetic evidence for genetic mutations, for cancer, or for toxicity to the embryo. But what we are discussing here are epigenetic events which one cannot conclude from the standard testing about the danger of a particular substance with the, what I call it, standard testing systems. You might keep in mind also that in addition to the developmental effects, that adult exposure to the same agents that have been exposed during development may add fuel to the fire in the same way that the hormones of puberty add to the DES story, or the postnatal administration of estrogens will intensify what results from the neonatal exposure. A last point, and I don't even know how to raise it except there's considerable interest in it, and that is whether there are multi-generational effects of this early exposure to hormones. Is the third generation going to show problems? Experimental data, and they are controversial, suggests that there may be, in some cases, third-generation effects from DES administration to a mother or to the second generation, which is the neonate. Workers in this area suggest that there may be also genetic effects of DES beyond its estrogenicity. And if it's true for DES, it could very easily be true for environmental estrogens as well. So, you've had my historical presentation of the background to our present concern. If DES affects humans and mice, and if other so-called weaker estrogens affect mice, maybe humans, we should worry indeed what environmental chemicals can do to humans and to living organisms generally. Thank you.
Howard, you made sort of an off-the-cuff statement here that the estrogenic load in the environment may be increasing. Now, the studies I've heard today, there are a lot of them that say we now recognize new effects of past exposures. We're seeing some long-range effects of past exposures. We still have a substantial burden of estrogenic xenobiotics in the environment from past and current exposure. But in view of the fact that some of the most estrogenic xenobiotics in the past were the chlorinated cyclic organics, and a lot of those are no longer, have been eliminated from use in this country.
Is the environment increasingly estrogenic? This is a matter of over decades. The changes that you talk about and replacements have occurred within the past really few years, and I don't know anything about the load as it may have changed then. The kind of things I was going to point out have to do with even the decreasing age at puberty. I don't know that this is due to environmental estrogens. I do know that it has accompanied increasing utilization of agents which could be estrogenic. The increased mortality from breast cancer, despite all of the efforts, it's very slight. It sure in hell hasn't gone down. And what was the third thing? The increase in ectopic pregnancies, which seems to me to be quite real and very clearly possibly related.
My real question is, okay, in the last 15 or 20 years, within this country at least, we made some steps to reduce our exposure to a number of the important estrogenic xenobiotics. Have we reversed that in some way by now adding more new ones so that we have an actual increase amount of new estrogenic xenobiotics going into the environment?
Well, I think Gates' point is very well taken, that the recognition of more estrogens does not represent an increase in the actual load to which people have been exposed. And I think there's a certain point that we have to recognize there. It's like with certain diseases whose incidence has been reported to increase, but really it's due to the better diagnostic methods. It's the same kind of a problem.
I guess there's one other point as well, and that is that there's a time lag in all of these things, the response. If in fact the lower sperm count is due to lower numbers of Sertoli cells, which were established during fetal development, and it's going to take one generation before you see a recovery, even if you get rid of all these chemicals. So we banned a lot of chlorinated chemicals in the early 70s. Endangered Species Act, Clean Water Act. You know, let's not let them change those particular things. But the exposure of children and fetuses in the 70s is what we're seeing now with decreased sperm counts as time goes on. The thing that Lou Gillette said was the best indicator of lowered sperm count is the age of the person or the date of birth. So we can get rid of all of these things and we can come back on a better plane, but we're not going to know we're on a better plane in terms of sperm count until the kids that are born today participate in these tissue banks, day after tomorrow for my kid.
My name is Bruce Boyd, and I work with the Regional Water Quality Control Board, and for the last 4 years up here, I've been coordinating the ESM and Muscle Watch programs, and also the Big Protection Program that looks at sediments. And I guess I'm one of those males at the water board that we've heard the deriding remarks about, but that isn't it. It's all ask you about that. I'd seen a video that several of today's speakers were in. It's a documentary, Assault on the Nail, and I believe you were. I believe you were in. But I thought Marion Moses was in there.
No, I don't know. I've been in a lot of things. I didn't see it.
We were not.
There was cell culture work done in that, in breast cancer research, and the discussion that was pursued in there was that there was a real hard time. First, procedures were done to isolate which part of the apparatus were causing the exposure that was bringing about the estrogenic responses in the cell cultures. And then there was the confounding issue of finding out what was in that particular culture tube that was causing the reaction. And my question relates to something that's been skirted around and mentioned, how hard it is to find out what is in a lot of the common products. Not just the stuff that we shouldn't be buying and spraying around, but the upholstery, the carpet glue, the wood, the water bottles, different brands of water bottles on the shelves, and that there isn't an obligation.
Did you say water?
Bottles. You can get bottled water and a lot of different.
Which have methylene chloride, but not anymore.
Yeah.
That's with CLA.
Yeah, that's right. That's better.
So I guess I'm looking if there has been any effort of publishing or making available to people in layperson's terms a way of assessing products, commonly sold products, for what they're made of and what the risks are of having them around.
What I'm asking for is still on the agenda, and I don't see it emerging. You may recall a slide that Dr. Moses showed you of, was it the EPA or something, with its many steps for investigating new compounds. And you might notice that with all of those 10 very sophisticated or what have you steps, there still is no concern for developmental effects. And this is really something I feel I have to reiterate, that I think these tests for detecting estrogenic activity in the environment are absolutely essential. But I don't think they answer the question about what the developmental effects of such agents are.
I guess to some that I'm hearing a similar answer from both of you is to develop methods to measure the functional effects rather than worrying about this chemical specificity. If it has the effect, that's causing it.
Yeah, I think we actually agree on this. We don't agree on anything else.
That's only because it's liquid.
Developing a cheap way of detecting the functional effect goes over a broad group of chemicals. Once you find the effect, then you can go and pinpoint, find the chemical, identify the chemical, and then hopefully legislate against it.